Single cells can move on their own by polarizing and generating traction forces, but often they coordinate with neighbors, enabling collective motion essential for processes like wound healing, development, and regeneration. In a recent paper, we studied the collective motion of cells using sphere-shaped mouse pancreas organoids. Videotaping the organoids revealed 3 modes of motion: persistent rotation with a stable rotation axis, orientational drift of the rotational axis, and rotational arrest. During persistent rotation, we found that cells are elongated either to the right or to the left relative to the rotation axis. Like right and left hands, these cellular patterns are mirror images of each other that do not match no matter how we rotate them, a phenomenon called chiral symmetry breaking. We used a vertex model of biological tissues in which cells are represented by polygons and accounted for cell traction forces to simulate rotating organoids. We first studied how cells align their traction forces with the neighbors. These simulations can explain the persistent rotation mode. However, the cell shapes do not reveal a chiral symmetry breaking as seen in experiment. What is missing in this model? We realized that we need to add a little bit of noise. Noise allows cells to wiggle and rearrange so that the tissue can flow a little bit on top of the solid body rotation. These flows are slightly different on the 2 hemispheres, which breaks the chiral symmetry, reproducing experimentally observed cellular patterns. Can we understand how these patterns appear? To find out, we looked at how traction forces align on the sphere. We found that they orient radially or in a spiral around each pole, forming so-called topological defects. The asymmetry of the defects reflects the chirality of traction force organization, which is present even without the noise. However, the cell shape pattern can only reflect the chirality of traction forces in the presence of noise. This work highlights the role of collective dynamics and topology in shaping chiral cell patterns in tissues. For details, check out our paper.